Hello students, this is day 2 of the RT-PCR Lab. Now, if you recall, yesterday what we did, we took our tube and made some cDNA copies of all the mRNA our mouse liver would produce. Just to review very quickly, we've got our tube here and we have thousands and thousands of different copies of mRNA, all types of mRNA doing different things. They're making all kinds of different enzymes, all kinds of different proteins. We didn't specifically select any mRNA. All we did yesterday was to add a whole bunch of tails of T's, so they would bind to the poly A tails of all these mRNAs that's in our test tube, and then we allowed reverse transcriptase, an enzyme, to go backwards along here. It binds to these poly T tails, and then it runs backwards and makes a cDNA copy of the mRNA. So remember, we made some hybrids. The mRNA is up at the top, and the reverse transcriptase made cDNA, which are DNA strands without the introns. We have no idea what the introns were. All that's left in the mRNA is the exons. So it's not true DNA, it's cDNA, and that's what we've made. But of course, the name of the lab is RT-PCR of the GAPDH gene. So we want the GAPDH gene. So up here in our test tube, we have the GAPDH gene, but we also have thousands and thousands of other copies of other mRNA that we don't want. We want to get rid of all the other mRNA, and we want to specifically select for just the GAPDH gene. So that's what we're going to do today. Day 2 is all about amplifying the GAPDH gene and not amplifying any other segments of DNA that do not contain the GAPDH gene. So here's what we're going to do. We're going to add a little bit of our master mix at our lab stations. So what we did is we created a master mix that contains all the buffers, primers, enzymes, and nucleotides necessary for our PCR reaction. We put it all in one tube. That way it's easier to aliquot it out and we don't accidentally leave anything out. The really important thing that is in this tube, though, is right here. The primers. The primers in this tube, what their job is to specifically target the GAPDH gene. They select only for the GAPDH gene. These primers will not bond to any other gene that is in our DNA except the GAPDH gene. And we know how it does that. Here's the cDNA strand that we made, cDNA, and along the cDNA we have a very specific sequence of nucleotides. It could be A, C, T, T, A. It doesn't matter what it is. The key is that it is very specific, and it comes just before what we want. In this case, right before the GAPDH gene. So just before the gene, we have this very specific set of nucleotides, and we can take advantage of that. We can actually call a biological supply company and give them this specific sequence of nucleotides, and tell them we would like a primer that binds to that sequence. And that primer will attach to that sequence of nucleotides and amplify the GAPDH gene. So if you think back to our transcription translation talks, we need the complementary strand to match this. So our primer that we have in the tube is going to be a sequence of nucleotides, usually 20 to 30 base pairs. In this case, I didn't draw 20 to 30 just to save some space. So let's draw the complementary strand. Remember, it's DNA, so we're using T's instead of U's. So this here is the primer that we've purchased, and it will bind only to the sequence just before the GAPDH gene. And then what we're going to do, let's go back over here. The second most important thing that's in our tube is here, enzymes, specifically our Taq polymerase enzyme. You should remember this word, polymerase. This should sound very familiar, like DNA and RNA polymerase. And in this case, Taq is simply a generic DNA polymerase. So this will make DNA. So this Taq polymerase, it will bind onto our primer, and then it will go down the line, and it will insert nucleotides, and it will make a new DNA strand using the primer and copying the GAPDH gene. It will be the complementary strand. We also have a primer going the other way that will make a copy going this way. That's a little bit more advanced, and we can talk about that at a later point. But today, all we're trying to do is separate out everything that is not GAPDH. So let's say this is our GAPDH section of cDNA that we want, and we get rid of everything else that doesn't code for GAPDH gene. And that's what today is. So we're going to add all of this stuff to our PCR tube. Notice we're going to save our single-stranded cDNA. That's our tube from yesterday. So we're going to add a little bit of our tube from yesterday, the single-strand cDNA, and the PCR master mix. That's the tube we're going to use today. We're going to store the one from yesterday in the freezer, and then we're going to run a PCR cycle. And then we're going to run a couple more. Note we run 34 cycles to amplify the GAPDH gene. So what happens is every time we run a new cycle, this Taq polymerase will run down here and make another copy of DNA. But now we've got 2 strands that we can copy. The Taq polymerase will now jump on 1, jump on 2, and make 2 copies. So now we have 1, 2, 3, 4 copies of our gene sequence. And Taq polymerase can use all 4 of those as a template. So very, very quickly we will have exponential growth going on. We will have exponential copying of DNA. So after a few cycles, after 7 or 8 cycles, we will have millions and millions of copies of our DNA strand. All of this original stuff, and this is very important, all of this original stuff is still in the test tube. We do not destroy it, and it does not go away. But when we run it on a gel on day 3, there might only be 1 or 2 strands in there, and the gel is not sense enough to pick that up, so we won't see it. What will show up, though, is these millions and millions and millions of copies of just the GAPDH gene. And that's what we're going for, and that will be day 3. So day 2 is really just adding a few things to the tube, and then the PCR machine does the hard work of 34 cycles. And then it will hold our tubes, hold them at 8 degrees Celsius, and it can hold them like that for a really, really long time until we go to day 3 and we check our results on a gel. We'll run gel electrophoresis on day 3, and that'll be the next video.